title your notes, Cellular Respiration. A couple things we're going to say about this reaction is that it is when chemical energy is used to make ATP. ATP is the universal currency, quote unquote. Taylor, do you mind shutting that door for me? Right over there. Thank you. Quote unquote universal currency. And what all that means is that it's no matter where you are in the cell, ATP can be used. It can be used over here, any organelle, any part of the cell. In fact, any cell in the body is also going to be able to make use of ATP. So ultimately, we have to get all of our chemical energy made into ATP. Universal currency of energy. So this cellular respiration is composed of enzyme catalyzed reactions. So this is also, you guys, why we spent so much time talking about the enzymes, is because they're the molecules that make this happen. Enzyme catalyzed reactions. They're usually catabolic, okay? If you remember what catabolic means, it means that you're taking a larger molecule, breaking it down to liberate the energy. So these are enzyme catalyzed reactions that break down organic molecules. In our case, we're going to look at sugar, and it'll pretty much be the only one we talk about. But there are metabolic reactions that will also break down fats and break down proteins. Have you guys ever heard of the new there's kind of this movement of it's like a diet thing, but it's ketosis. Like you're supposed to try to get your body to go into ketosis. Yeah, so the idea there is that if you reduce your carbohydrate intake, your body will kind of stop doing that kind of metabolism. And that's the metabolism that breaks down sugars and gets your energy from carbs. But if you stop the intake of carbs, your body has to go this way and do other reactions that derive energy from proteins and fats. That's kind of this idea with this diet. And the reason that the reason it's so popular is because we really believe that especially the American diet, but the modern diet, let's just say modern diet, is way off balance. Like we're consuming way too many carbs. Like it's out of balance. The ratio of carbs to protein and fat is like skewed. And so the idea is that that's probably causing health problems, and we need to get it balanced, and we need to do more ketosis. So anyways, I talk about the ketosis because those are the metabolic pathways that I think it's protein primarily that is processed with those reactions. The sugars and the carbs is a different pathway, and the sugar carb pathway, that's the one we're going to look at right now. Okay, now it's time for the big picture. Do you guys still have room for the big picture? You might want to turn your page sideways. If you don't have enough room, maybe go to the next page already. I don't know. But we're going to have the big cell, and the cell is going to do this. It's going to take up like the whole page because we need a lot of room to show all the moving parts. So this, what we're going to look at today is the aerobic. This diagram is going to be of aerobic cellular respiration. And the equation to summarize what's taking place is this. You've got C6H12O6, that is a glucose molecule. plus 6 oxygen molecules. So this is why you got to make sure that your mesocosm is getting oxygen. If it's not getting oxygen, your animals are not going to be able to do cellular respiration. And then what's going to be produced is 6 carbon dioxide molecules. And this is why your mesocosm must have a way of absorbing carbon dioxide. Because what would happen, you guys, if we sealed up, like let's say we tried to survive in this room for like a month and there was no circulation, like the vents were closed off and it was just sealed up, we wouldn't be able to survive. The carbon dioxide levels would begin to rise to toxic levels and we would suffocate. That's what would happen to your animals in your jar. If you close the lid and you seal up your jar, they will suffocate unless there is a reaction that is going to process the carbon dioxide. And it's a very famous reaction. So don't worry. Our plants are going to process the carbon dioxide. Hopefully. So that's our overall reaction. Remember, you guys, what we said, this is what we're after. This is the whole reason we do cell respiration. We're after that ATP. Without the ATP, your body falls apart. Can't do anything. And remember what we said before, that your body literally would fall apart. The second law of thermodynamics says that energy is required to keep you together, keep you from decomposing. So there we go. Now, here is the big cell. We're going to draw it like so. And it's going to represent an animal cell. So it's kind of roundish. There's no cell wall. It's just a plasma membrane. And so the very first step that has to happen is we have to bring in a sugar molecule. So picture a little glucose transporter. We've got a little sugar molecule here. The sugar molecule comes in. So this is like a little glucose, right? Glucose has to come in. By the way, the glucose was probably in a capillary. It was like there's like a little blood vessel or a little capillary that little capillary delivered the sugar, and then it was absorbed into the cell. So what happens next to the little sugar that's now in our cell? The very first, so right now it's in what we call the cytosol. The cytosol is just the liquid inside the cell. The glucose molecule will go through a reaction called glycolysis. Now, students, just a little warning here. I am oversimplifying most of these reactions. So I'm telling you right now, I'm not showing you all the steps because I don't think it's necessary. I think that if I included all the steps, it would overcomplicate it. So this glycolysis reaction essentially splits the sugar into 2 pieces. Can everybody kind of picture that? Like if you tore the sugar into 2 pieces, you would get 2 pyruvates. Now again, students, the tearing of the molecule into 2 pieces, not entirely accurate. It's a little more complicated than that, but you get the idea. So 2 pyruvates are produced during glycolysis. Okay, so now we have the mitochondria. The mitochondria sits over here, and I'm drawing it enormous. The mitochondria is not drawn to scale, but we have to draw it large because we got to fit everything in there. So remember that the mitochondria has an outer outer layer and an inner layer that folds in. These cristae, as they're called, they help to increase the surface area of the mitochondria. Now I'm drawing it kind of funny over here on purpose because I'm creating some space for us to draw some stuff. But remember that the mitochondria, the folds, they help to increase the surface area, thus giving more surface for these reactions to take place. And that basically just increases the efficiency of the cell, of the organelle. Okay, so that's the mitochondria. And if oxygen is present, and that's what we would call aerobic conditions, right? Aerobic. If oxygen is present, the pyruvate will go into the mitochondria. So you can add it there. There's the pyruvate. It's going into the mitochondria, and this is where it's going to be processed. Okay, so the pyruvate is then made into acetyl-CoA, and then the acetyl-CoA is sent into the Krebs cycle. Krebs cycle. And the Krebs cycle releases carbon dioxide, and it also releases 2 ATP. So you can go ahead and put 2 ATP, and then you could put CO2 waste. The reason I call it waste is because it's toxic. If the CO2 is not released, it would become toxic to the cell. All right, so we got to get rid of it. And then I did kind of forget something, students. Back up here, the glycolysis. This catabolic reaction that we call glycolysis does produce a little bit of ATP. So you're going to have your adenosine diphosphate, and then plus 2 phosphates. So you can think of this as like an ADP connected phosphate, another ADP connected phosphate, and then what you're going to get out of it is 2 ATPs. And again, that's what we're after, right? The whole purpose of cell respiration is to produce the ATP. So we're doing good. So far, we've made 4 ATP. It's pretty good. But we can make a lot more if we do some more work here in the mitochondria. All right, so back to the Krebs cycle. So we've said that the pyruvate gets converted to acetyl-CoA. The other thing that the Krebs cycle does is it energizes some energy carriers. So there's an energy carrier name called FAD+, and there's another energy carrier called NAD+. And what the Krebs cycle will do is it will add electrons to those energy carriers. So let's, we're going to show that here. So the energy carriers come in to the Krebs cycle. They get an electron added to it. And so then they become, if you add an electron to FAD+, it becomes FAD H2. And then that reaction will turn NAD plus into NADH2. I'm sorry, NADH. There we go. All right, so just conceptually, what I want you guys to picture here is that this is the unenergized version. This is like the spent version. It's already spent its energy. And so what it has to do is it has to come to the Krebs cycle to be re-energized and turned back into these molecules. These are the energy carriers. These are spent energy carriers. So just kind of picture the flow of energy, right? We got this, a lot of energy stored here. The energy is now kind of, some of the energy was put over here. Some of the energy was given to these ATPs. And now the energy is being, the leftover energy is being given to these guys right here. So you can kind of follow the energy and where it's going here. So these energy carriers will now carry the energy to another part of the mitochondria. They're going to flow over here. So there's some proteins that are embedded in the inner membrane. So you need to know that this is the inner membrane. In fact, you're going to get some questions on the test that focus on the inner membrane of the mitochondria. So I want to underline that for you. The inner membrane is very important. But anyway, we got these proteins that are embedded in the inner membrane, and they're going to help us get a whole bunch of ATP. I will show you how. Okay, so here's how they do it. The energy carriers will move over here, and they're going to release protons. Into this inner membrane space. Is everybody okay with that? Because you see here, right, that it gained a proton, and now it's taking that proton and transferring it into here. And so what do you get if NADH gives up its proton? Well, then here's what we have left, right? We're back to NAD plus. And then, of course, that NAD plus will come back over here, and the process repeats. So it's a cycle. There's a cycle there. Everybody doing okay? This is no joke. I mean, this is biochemistry. You know, it's tough. All right, so we know now that the energy carriers have released their energy into this inner membrane space, and we're building up an accumulation of protons. Big accumulation of protons. So let's draw some more. Let's draw some more protons, just to kind of illustrate the point. Big giant gradient. Okay, so that's good. There is another product here. If we donate the proton, we're also going to have an extra electron that needs to be dealt with, and that extra electron is going to be received by oxygen. So we're going to have this little electron is going to come over here, join up with an oxygen molecule. And then, and this is the reaction. The oxygen is going to have the electron plus 2 protons join up with it, and it's going to produce a water molecule. So what we've got down here is oxygen acting as a final electron acceptor. Do you guys remember our discussion about oxygen being electronegative? You guys remember that? This is further proof that oxygen is electronegative, right? It has an affinity for electrons, so it becomes a really good electron acceptor. So the oxygen atom, or molecule, is acting as a final electron acceptor. Okay, so we gotta pause here for a moment because we there's a lot going on, and I want to show you that the products and reactants of our original equation. If you look at the top, right, we had this original equation. The products and reactants have been mostly accounted for already. We're gonna look at that in a second. Okay, so there's one more step here. Oh yeah. Okay. So this process of the electron being received by the oxygen molecule, this is called the electron transport chain. Electron transport chain. Underline that because I want to make sure you guys know where that is and what that's referring to. All right. Okay, so the last step is we're going to try to get every ounce of ATP that we can. All right, we're trying to get as much ATP out of this as we can. And by the way, this is so far we've only processed one sugar molecule. So this is what takes place just to process one molecule of glucose. So the next step is something called chemiosmosis. And that is when all of this buildup of protons, this gradient of protons, is going to be let in. We're going to let it in, right? So here come the protons. They flow from an area of high concentration to an area of low concentration. And they flow through a protein. I'm going to label this little protein here as ATP synthase. Now, this protein that we call ATP synthase, you can probably guess what it does just based on its name, synthase. It's going to make ATP. So how much do we get from this reaction? And by the way, the reaction is called chemiosmosis. So I put that in parentheses because that is the reaction that takes place with these embedded proteins. Okay, chemiosmosis. Okay, so here's what it does. It takes 36 ADPs plus 36 phosphates, and it makes 36 ATPs. So that's a lot. That's a lot more ATP than we saw anywhere else in this process, right? In other words, most of it is coming from the chemiosmosis. And it relies on this proton gradient. In other words, if this proton gradient doesn't exist, we're not going to get much ATP. Very much relies on that. All right. So as promised, I told you that this big reaction is a summary of this equation at the top. Let's confirm. Let's confirm it, because I want to check that we've got all the inputs. I want to check that we've got the outputs all accounted for. So let's start with our first input, sugar. Did we show sugar going into the cell as an input? Yes. Check, check. That's right here. Sugar going in. I'm going to check that off. Did we show oxygen as a product? In other words, oxygen has to go into the cell to get consumed. Where is that? Down here. Oxygen coming in, right? Has to diffuse into the cell in order to become the electronic acceptor. Check, check. Did we show carbon dioxide as a product, as a waste product? Yes, right here. It is a product of the Krebs cycle. It's produced. So we check that off. Did we show water molecules as a product, something that's made during this process? Yes, it is part of the electron transport chain. It is the product that we get when oxygen receives the electrons and protons. Check, check. And lastly, did we show, by the way, I think the number here, what did we get? 36 plus, I don't know. I think you're supposed to yield 38 ATP. I'm not sure about that number, so maybe you don't quote me on that, but you're not, don't worry about that. You're not going to get asked how many ATPs are made, but I think it's 38. All right, did we show ATP being made? We did. We've got that down here. In fact, there's a couple places. There's some that's made here, there's some that's made here, and then there's a whole bunch that's made over here. Okay, so students, did I just show you aerobic or anaerobic respiration? Aerobic because oxygen is one of the reactants, right? But what if we don't have any oxygen to react? Or does cellular respiration just not happen at all? Or is there an alternative pathway that we can use? So it turns out that if oxygen is not available, we can go this way. So we're going to say if O2 is absent, and that would be called anaerobic, then we're going to do what we call fermentation. And there's a couple things that can be produced. If it's an animal, you're going to produce lactic acid. So that's usually in animals. If you're a yeast cell, so if you're a yeast, you'll produce alcohol, and that's how we get like beer and wine and stuff, right? Beer and wine is produced using yeast fermentation. Basically, like if you got a big old vat of grape juice, is basically how wine is made, right? And then you just pitch a whole bunch of yeast cells into there. What will happen is the yeast will consume all of the oxygen. It'll do aerobic respiration for maybe an hour or two. And then after that, the oxygen is completely gone. And the yeast go into anaerobic respiration, and they consume all the rest of the grape juice doing fermentation. And so one of the products of the yeast is alcohol, and that's what makes wine alcoholic. Lastly, we do get a small, I gotta say smaller amount of ATP. I know it's a little bit vague. Sorry, I don't know how many ATPs you get from fermentation, but it's less. In fact, it's much less than you would get if it was aerobic. All right, guys, any questions about this? You guys got it all memorized? All right, good. We got a quiz on it right now. I'm just joking. All right. Ha 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 ha.
What into the FADH like see as the product like NAD plus like that?
Oh yeah. Okay. Sorry about that. I don't really have room to write it. I just assumed you guys would also know that if it wasn't if it was the FAD, or I'm sorry, if it was the FADH, it would do the exact same thing, and it would just be FAD plus, and then FAD plus would come back around, get re-energized, and the cycle repeats. So students, you got to have the eyes for the energy. I want you to be able to trace the flow of the energy. So energy, there's a whole bunch of stored energy in this molecule. Some of the energy gets liberated here and captured here. Some of the energy gets liberated here as ATP, and then some of the leftover energy gets passed on to these molecules, which then deliver their energy over to here, and then that energy gets added to this energy. So you're kind of following the energy as it goes. And the general rule, as we said, is that as molecules get broken into smaller and smaller pieces, more and more energy gets liberated. It's this idea of free energy, right? It's a second law of thermodynamics with entropy and all of that. Any other questions? Okay. We'll go ahead and stop the video.